So thank you very much again for inviting me to give this talk. I'm really interested in your feedback on it actually, but also hope it shows you just how useful metabolomics has been to us within the diabetes in pregnancy field. So first of all, I am a professor of chemical pathology or medical biochemistry and diabetes in pregnancy, and I recently moved from Cambridge to Leicester. And for those who are outside of the UK, Leicester is more or less right in the middle of England in the Midlands. So just to start with a little disclosure slide. And then we'll move on. So, research in my group studies maternal metabolism. So, what actually happens in diabetes in pregnancy? And so, our overall aims really are to look from that period from conception all the way through pregnancy to postnatal health for mothers and children. And we do that with a whole range of clinical studies. And overall, our aims are to identify the causes and consequences of diabetes in pregnancy, to understand the effects of diabetes in pregnancy upon long term health, both for mothers and for offspring, but also to translate our scientific discoveries into meaningful clinical advances. So, why study diabetes in pregnancy? Well, there's three main types of diabetes in pregnancy. There's gestational diabetes, by far the most common, but there's also type 1 diabetes and type 2 diabetes, which are in general more severe because they've got higher glucose levels. Now, despite the difference in severity of glucose levels, actually there are common complications affecting all forms of diabetes in pregnancy. And one of them, for example, is big babies or macrosomia. So macrosomia is a term that refers to babies over around 4 kilograms in size or large for gestational age would be greater than the 90th centile. And this is a really common complication in women with all forms of diabetes in pregnancy. Now, this has, of course, huge burden to individual patients and to their children, but actually it also has quite an important health service repercussion. So we know that healthcare costs really increase in women with diabetes in pregnancy. During the pregnancy itself, but actually up to sort of 7 or 8 years after pregnancy. We also know that each baby that's born bigger than average or bigger than normal, macrosomia, has quite a degree of additional cost to the health service. And of course, there are increased risks, for example, of perinatal mortality in babies, slight rare but important all the same, and other complications as well. Another good reason to study diabetes in pregnancy is that actually the international burden of diabetes in pregnancy is extremely high and likely to rise over the next number of years. And so this is a graph, this is a picture looking particularly at gestational diabetes, but you can see that actually prevalence does vary from Europe, 6.1% to sort of 15% in Middle East and North Africa, and also 15% really in Southeast Asia. So this is a huge amount of pregnant women every year across the globe who suffer from diabetes in pregnancy. And really, the third reason for studying diabetes in pregnancy are that we know that it causes both short term and long term health sequelae in offspring. So, we know that intrauterine exposure to maternal diabetes has consequences upon that perinatal period in terms of perinatal complications like large for gestational age, neonatal hypoglycemia, which is where babies reduce their sugar levels after birth in response to the hyperinsulinemia they've developed, shoulder dystocia, which is where sort of large or chunky shoulders prevent successful vaginal delivery, and that can be very dangerous, and also respiratory distress. So babies can struggle to adapt to that postnatal environment. But looking through infancy towards childhood and adolescence, there's also increasing awareness that intrauterine exposure to maternal diabetes creates longer term consequences. For example, we know that children and adolescents exposed to diabetes in utero have abnormal glucose handling themselves in later life. We know that there's evidence of more insulin resistance in childhood and adolescence, increased risks of obesity, and while the long term studies really have yet to be done looking at adult metabolism, we expect that those childhood and adolescent risk factors culminate in a profile which really predisposes to insulin resistance, obesity, and type 2 diabetes in adulthood. And of course, these are potentially people who develop type 2 diabetes earlier because of that lifelong predisposition, resulting in really considerable degrees of multimorbidity. So, where do we go from here? Well, one of the things that we've been able to do is look for the first time in much more detail regarding how these complications develop. As you can imagine, it's actually really difficult to study human pregnancy. It's really difficult to get a very good grasp on what's happening in that situation. There's very limited ways we have of observing metabolism in the womb. However, it is really important that we do that. And also, of course, it's really complicated because there's essentially three different aspects. There's the maternal aspect, there's the fetal aspect, but there's also the placenta that's doing its own thing, which also we really struggle to study in humans during an ongoing pregnancy. So while we can take placental tissue at the end of pregnancy and we can also take cord blood from the umbilical cord, we actually have very little way of really understanding what's happening in that interplay between those three systems: mother, baby, and placenta. So, one of the things we've been able to do then is to use maternal serum as we go through pregnancy in trimesters 1, 2, and 3, and using the metabolomics to guide us as to the looking at changes in metabolism through pregnancy. So, for example, we did this first of all for big babies. So, this is actually extra large for gestational age babies. So, with a birth weight over the 97.5th centile. So, in normal circumstances, in a normal healthy population, only a very small number of babies would be at that level. But actually, within a type 1 diabetes situation, actually lots of babies are born over the 90th or over the 97.5th centile. And you can see here that we have used our metabolomics to assess at 12 weeks, 24 weeks, and 34 weeks what is happening within the maternal circulation. And you can see that actually, sorry, did I just go on too quickly there? There's 12 weeks, 24 weeks, and 34 weeks. You can see that actually at 12 weeks, our log P here for significance is 3. There's very few elements, very few metabolites that are significantly different when we compare pregnant women who went on to have an extra large for gestational age baby compared to women who didn't. And that shows us that really not a lot is happening at 12 weeks in terms of the metabolism that is significantly different. However, as time goes on through pregnancy, 24 weeks and 34 weeks, you can see that there are changes. So, looking at these, there's the metabolite class along the bottom, which I've given lots of different colors to. And then this is the degree of significance or minus log P. So if a minus log P is above 3, so in this section, then there's a significant difference in that metabolite between women who had a large for gestational age baby and those who didn't. So you can see that actually most of the changes in association with large for gestational age are really happening in that last period, that last trimester of pregnancy. And this is really helpful actually for us to know. Partly it helps us target when we should be looking at babies in terms of their pregnancy care, but also it helps us if we're thinking about biomarkers for this condition, really actually probably there's not a lot to go on from 24 weeks. So moving on then to another complication, neonatal hypoglycemia. So this is a very different pattern, and that in itself actually is really interesting. So before this study, the general belief within the diabetes and pregnancy field was that babies who had too much glucose exposure in the womb produced too much insulin, so hyperinsulinism in pregnancy, and that caused the babies to grow larger, but also to have low blood sugar levels after birth. And you can see from this slide actually that the pattern, however, is very different from the extra large for gestational age babies. You can see here that there's really pronounced amounts of triglycerides from 12 weeks, which occurs at 24 weeks to some degree as well, and also is present at 34 weeks. So we were able to show for the first time that actually early pregnancy metabolism, while it may not have a direct influence on baby size, is actually really important for baby metabolism. And we're still investigating this aspect further, really, to see if this continues to be an issue in other cohorts. So moving on, preeclampsia, many people have heard of. It's a condition where there's high blood pressure in pregnancy, but actually preeclampsia also occurs more commonly in people with diabetes in pregnancy. And although preeclampsia often occurs around about 32 weeks or so, in practice, actually, it can often be present a little bit earlier. And what's interesting here is that there are changes in the metabolites all through pregnancy, even from the first trimester, in women who go on to develop preeclampsia. And I think this is really interesting as we think through, how there have been a number of successful biomarkers for preeclampsia outside the diabetes population, and would it be possible for us to be using some of our data to look for a more tailored biomarker in diabetes? Quite possibly. So moving on then to think then through really the next part of the conundrum. So how does exposure to maternal diabetes cause the longer term health sequelae in offspring? So we've talked already a little bit about these short term perinatal complications. So we can show very clearly that there's altered metabolism in pregnancy, predisposing to or contributing to the pathophysiology of these disorders. But actually, what about these long term disorders? How is it possible that a short period of altered environment, if you like, in utero can actually translate into a long term change in adult metabolic risk, really? And so, from my point of view, I think there's two main ways that this could happen, which are, of course, interrelated. And one is that the programming effects of early life upon body composition and lipid homeostasis could well account for these long term differences in outcomes in adolescence and adulthood. However, we really lack those long term studies looking at this. You really need to study a huge number of people in pregnancy to get good follow up through life to get to adolescence, let alone adulthood. So, from that point of view, it's really difficult to do these studies long term. But actually, do we have any clues that we could use to assess whether this is a reasonable hypothesis or a reasonable mechanism of action for this effect? So first of all, we know that maternal high glucose levels or hyperglycemia does directly influence offspring body composition or is strongly associated with body composition in type 1 diabetes. And I'm showing you data here, both the centiles here in pink, but also from the skin fold sum. So when babies are born in our studies, we do skin fold measurements. It's like a pinch of their fat levels, which is measured and used as a sum to assess the sort of total fat availability really in the baby subcutaneously. So you can see here that maternal glucose, and this is time above range on continuous glucose monitoring, and we'll go into that in a moment. Time above range, you can see, was associated with centiles, but actually even more strongly with the degree of skin fold sum, so the degree of fat in the baby's body at birth. And we've had other encouraging evidence really to point that this direction from our work in gestational diabetes. So this is a study that I was involved with a few later, if you like, a study called Upbeat that was run in London quite a number of years ago, looking at whether or not we can prevent gestational diabetes. And we did a secondary analysis of that data, looking at the lipidome, and you can see that actually women who developed gestational diabetes had really high and pronounced levels of many different diglycerides and triglycerides, which were strongly associated with de novo lipogenesis. And so, lipogenesis is the process where new fat is made. And typically, of course, new fat is made from excessive glucose or carbohydrate or even alcohol in the bloodstream. But women here with gestational diabetes who are having very little alcohol in general, we're showing this exaggerated degree of de novo lipogenesis. And interestingly, we were able to show that actually triglycerides that tended to be more from dietary sources, from lipolysis of standard adipose tissue, or total diet triglycerides were not influenced in the same way. So this led us to think that actually there is clearly something in diabetes and pregnancy related to lipid metabolism that may be important to study regarding the programming of offspring body composition later in life. So that leads me on then to our hypothesis. So our hypothesis then was that the effects of maternal hyperglycemia on offspring adiposity are solely mediated by altered maternal and fetal lipid metabolism in pregnancy. So that instead of a direct effect of maternal hyperglycemia upon offspring body composition, we actually have, if you like, indirect effects through altered maternal and offspring lipid metabolism. Now, again, this is a very difficult thing to really study directly or experimentally in humans, but we were able to use the metabolomics assessment of our CONCEPTT study to study this in more depth. So, the CONCEPTT study, as Alessandro mentioned, is the continuous glucose monitoring in pregnant women with type 1 diabetes trial. And this was a multi center randomized controlled trial published in the Lancet in 2017. And during this study, women were randomized to receive either standard care or continuous glucose monitoring in real time that they could use. I'll talk a little bit more about what continuous glucose monitoring is on the next slide. But for the purposes of this study, we also took maternal blood at three time points in pregnancy: 12, 24, and 34 weeks. We were really fortunate to be able to get cord blood at birth. There was about 225 women in the study at this point. We were fortunate to have cord blood on around 93 infants, which is a fantastic rate. Actually, they're very difficult samples to get hold of. And we were fortunate that Metabolon were willing to do the metabolite analysis, but also the complex lipid pathway for us. So, in terms of analysis, this was done in tandem by two investigators separately. We started off with the principal component analysis and partial least square discriminant analysis, but the results that we actually presented, because pregnancy is such a complex time, we presented them as unadjusted and adjusted, linear, and logistic regression. We use a modified Bonferroni method to adjust for multiple testing with a limit of significance of p equal to 0.001. The modified Bonferroni method we find very useful because the standard Bonferroni method does not allow for the interconnectedness of variables. And actually, of course, a lot of lipid species and a lot of metabolites in general are very interdependent on each other. So, what then is actually continuous glucose monitoring? Well, many of you will have been familiar with this. It's been quite widely mentioned in the media and so forth. So, continuous glucose monitoring involves having a little sensor placed underneath the skin. This is one example. And the sensor is able to read a glucose level in the interstitial fluid around about every 5 minutes. And that leads to around 300 interstitial glucose levels a day. It is, however, calibrated to venous glucose. So overall, it should be fairly similar to results from the capillary glucose testing. Uh, it's not as accurate as other methods such as the YSI reference standard for glucose, but actually from a day-to-day basis, it is extremely useful for patients to use to view their diet really and to make changes. So you can see that actually this tracing here is highlighting areas, for example, here, when the glucose levels are going a bit high. And over time, actually, people are able to change their diet and get a profile much more like this, where actually they are eating, but the glucose levels aren't going above target afterwards. Now, how do we actually analyze continuous glucose monitoring data? Well, there's a set of kind of standard CGM metrics. There's the mean, so mean CGM glucose. It's a time in range, time above range, time below range. We also use the coefficient of variation and the standard deviation of CGM glucose. Now, there's a range of ranges available depending on who is doing the CGM, but in pregnancy we use this range, which is actually extremely tight, so often very difficult for women to achieve, particularly with type 1 diabetes. So we have been able to use these metrics already in quite a number of other studies, looking at, for example, the effect of glycemia and how we measure it upon pregnancy outcomes. So this was looking at how we use various descriptions, both CGM metrics, but also biochemical markers of glycemia to look at large for gestational age for from 12 weeks. And you can see that actually overall the CGM markers are really quite good as predictors, perhaps not at LGA, but with other things, they're quite predictive of outcomes. So we're going to move on in the second section of my talk to talk a little bit about this more kind of focused hypothesis testing that we've been able to do using the metabolomics from Metabolon. But I'm going to hand back to Alessandro briefly at the moment, just so that he can update us with some other content.
Thank you very much, Professor Meek, for that lovely first part. I will now take the opportunity to review the Metabolon platform and introduce some of the products and services that we have available and that can be readily applied to enhance your research. So, in addition to the fascinating work you're hearing about today, metabolomics really has applications across a broad area of research fields and really helps in overcoming traditional challenges of translational research. For example, metabolomics can be used to discover and identify novel markers of disease progression and severity. And these markers naturally become druggable targets, where again, metabolomics can be used to assess treatment efficacy or identify secondary off-target effects, so address safety. But metabolomics can also be used to predict disease and disease outcomes. For example, our platform has been used to successfully build predictive models addressing COVID severity. But it doesn't end here. Metabolomics can be used to understand the function of our microbiomes. Metabolomics also enhances patient stratification in clinical trials, as well as adding insights into the understanding of functional genomics, ultimately linking the genotype to the phenotype. For example, looking at the effects of rare gene variants in different conditions. So I would say today, metabolomics should be one of the most important indispensable research tools in your research toolbox, and it is truly applicable at 360 degrees, allowing researchers to address a plethora of biological questions. Now, as we all know, tackling challenges and moving the needle in translational research really requires access to consistent, high-quality data and insights. And Metabolon approaches this challenge with multiple panel offerings, ranging from the broad discovery applications of our global discovery panel through to the targeted panels and even pinpointing single analytes. And these panels have been designed to answer different types of research questions, as well as address different application needs. Our Global Discovery Panel is our approach to untargeted metabolomics and offers unrivaled coverage using our proprietary library of over 5,400 metabolites. Untargeted metabolomics is a comparative approach, and our Discovery Panel offers relative quantitation and is ideal for getting the most data from your biological samples without specific knowledge of what biochemicals might be present. With this panel, we can capture changes relative to a variety of factors, including our genes, the exposure to the environment, diet, therapeutic treatment, and disease. So delivering the best possible representation of the phenotype at a given time point in time, so a snapshot. We have also recently launched more focused discovery panels tailored for researchers primarily interested in biological stress, kidney or liver function. And these panels also report relative quantitation of a subset of relative metabolites, markers that we have selected for you based on our institutional knowledge, and that come with a suite of advanced data visualization tools specifically designed to enhance interpretability of these large data sets for researchers. And if you look to the right, there are a few examples of our targeted and single analyte assays designed to build on and complement our discovery work. These offer absolute quantification of metabolites that have previously been identified as important in specific biological pathways or physiological responses. We currently offer 22 off-the-shelf targeted panels and assays, as well, importantly, as the ability to entirely customize panels based on your specific research needs. So if you have any specific markers you want to study, let's talk. Now, Metabolon truly sets the standard in metabolomics analysis, excelling in coverage, identification, and data quality. The main driver that enables us to offer the quality of results that we do is our extensive reference library composed of authentic chemical standards. And these standards have been run repeatedly on our platform, which has enabled us to characterize their exact molecular fingerprint. As I mentioned previously, our library covers over 5,400 metabolites and allows us to offer the highest level of annotation confidence, which is what you see on the left. So tier 1 and tier 2 identification, as well as depth of coverage. In fact, as you can see on the right, we capture metabolites from over 70 metabolic pathways on our global discovery panel, allowing us to provide insights into the multitude of diverse and complex interactions that contribute to a complete biological system. Now, what you see in this slide is our modus operandi. So don't worry, Metabolon is with you every step of the way, providing guidance to power your study for success all the way through expert analysis, interpretation, and data validation and visualization. We like to work with our investigators early in the process to ensure the study design and sampling handling protocols will deliver the best possible results to answer your research questions. Once the samples arrive in our lab, we perform extractions with unique SOPs based on sample type to prepare for analysis on our platform. Now, because the matrices we work with and analytes we cover are extremely diverse in terms of their biochemical makeup and physicochemical properties, this presents a great challenge in being able to accurately and repeatedly detect this broad range of metabolites. And to address this challenge and fully capture this biochemical complexity, we use different and complementary data acquisition methods designed to maximize metabolite detection and coverage. Once we complete this step, the data is then matched against our reference library to turn tens of thousands of ion features into identified metabolites that meet our rigorous and stringent QA and QC requirements. The final step of our pipeline is data interpretation and bioinformatics, where the identified metabolites are mapped to known biochemical pathways, so we can take you beyond a data table and elevate meaningful biological insights that help narrate a comprehensive and credible story of your research discoveries. Now, as I alluded to on the previous slide, mass spec data is very rich in noise, artifacts, and redundant signals, which may or may not be important or meaningful to the study. So how do we extract biological meaningful data and significant metabolites from these extremely noisy data sets? Well, at Metabolon, we use a chemocentric approach. So the projects we run can generate in excess of 150,000 ion features, but to be clear, 150,000 ion features do not translate into 150,000 metabolites in our samples. So we take these features and search them against our annotated reference library of pure authenticated standards, measuring against retention time on our columns, mass, and fragmentation pattern for each standard in our library. And this is where having a physical library of standards rather than a virtual one really makes a difference. Our library also contains signatures of the ionization products of every compound. And this allows us to filter through and remove noise and enable appropriate and accurate metabolite identification. This approach has the added benefit of reducing false discovery, which is a huge issue and a common pitfall of the big data that we see daily in the omics fields. Now, after the data reduction step to remove the noise and extraneous artifacts from our data sets, we are then ready to move forward with a confident list of identified metabolites to further interrogate for biological meaning. For our discovery platforms, we provide client data tables in multiple formats that allow researchers to directly import their data into their favorite environment tools, such as R or Python. For our targeted panels, we provide fully quantitative data for the specific set of metabolites of interest, which is also immediately usable for statistical analysis and biomarker validation. Our services do not stop at data generation. So beyond our client data tables, we also offer statistical analysis and visualization tools that allow researchers to immediately interpret key results, even without prior metabolomics experience. From there, we offer in-depth, fully referenced interpretative reports written by our in-house discovery and translational science team, who are able to take full advantage of their wealth of institutional knowledge, guiding you through key results and providing you with actionable insights. Now, before passing the stage back to Professor Meek, I invite you all to ask yourselves, what can Metabolon reveal for you?
Superb. So thank you again for that. We just to go back to what can Metabolon reveal and what can we learn from the metabolomics to understand the maternal fetal lipid metabolism. So we talked about the hypothesis really that the effects of maternal hyperglycemia on offspring adiposity are solely mediated by altered maternal and fetal lipid metabolism in pregnancy. And there's 4 arrows to this diagram, so we're going to look at the 4 bits in sequence. First of all, does maternal hyperglycemia influence the maternal metabolome? And the answer to that, it will not surprise you to know, is very substantially. So this is looking at maternal time above range or hyperglycemia on maternal blood samples at 24 weeks. And you can see that actually there's lots going on here. There's a lot of species over that line of significance. You can see particularly, I'm marking here the triglycerides and the diglycerides, both elevated. And we know that triglycerides actually in the bloodstream come from a range of different places, including de novo lipogenesis, lipolysis of adipose tissue, and diet. And we were able to use the Metabolon data to look much more in depth here. Now, this is a lot of information on one slide, but you can see actually the diglycerides, many of them have fatty acids 16 or 18 in them. And the triglycerides, actually, many of them also have fatty acids 16-0, 16-1, 18-0, or 18-1. So that's 16 carbon atoms, 0 double bonds, 16 carbon atoms, 1 double bond, and so forth. Those are the most common fatty acids to be made in lipogenesis. They're very basic fatty acids, the basic building blocks. And so we were able to show from this that actually these are in association with hyperglycemia. The body is doing what we sort of expected it would do, you mopping up that extra glucose to make new fat. And we could also see something really very similar at 34 weeks. So again, a similar range of fatty acids, also very much related to lipogenesis. So in answer to the first part of our hypothesis, if you like, how does maternal hyperglycemia influence the maternal metabolome? And it appears to be probably through making new fat or de novo lipogenesis. So moving on then to the next part, does maternal fatty acid abundance, for example, of de novo lipogenesis species, influence body composition in offspring? Now, we already were able to show, and we've shown this slide before, that actually maternal hyperglycemia in itself influences body composition and also, if you like, fatness or skin fold some. But what about the individual fatty acids? So looking at these individual fatty acids here, and also total fatty acids, monoacyl, diacyl, and triacyl glycerides, you can see that actually when we adjusted for multiple different confounders, including maternal glucose, you can see that there remain very significant associations between maternal blood free fatty acids and offspring skin folds. To some extent, in pink here with the centiles as well, but actually the most pronounced associations are with the skin folds. So from this, we can conclude that actually maternal lipid metabolism, and particularly fatty acid species in the blood, do appear to have an association, a strong association with fetal adiposity. So what about the next part of the puzzle then? How does maternal hyperglycemia influence the cord blood metabolome? Can we have a look at that? Now, there are fewer hits here because there are fewer samples, but you can see that actually there are still quite a number of species that fall above that line of significance. And particularly 2 sort of groups, there's the plasmalogens and also the carnitines. And you can see here that actually there were quite a lot of different kinds of carnitines at both 24 and 34 weeks. Now, carnitines are really strongly associated with the process of beta oxidation, so burning fat as fuel. And so, although we cannot directly quantify that in a fetus in utero, it does seem to appear that actually maternal hyperglycemia appears to prompt beta oxidation in the fetus, so burning fat as fuel. Now, this is actually extremely controversial because it's generally considered that actually the fetus relies on glucose only as a fuel, not fat. And we are looking into this further, but it's the first evidence really in humans to suggest that actually that may not be true in all cases. So while the fetus does rely on glucose a lot as a fuel molecule, it's of course totally possible that fetuses, and particularly in pathological situations, may turn to other fuels as well. So then moving on to our last piece of the puzzle. So then what about cord blood carnitines? Are they associated with body composition? Do we think the degree of beta oxidation might be relevant here? Well, actually, when we looked at a whole range of different carnitines, and again, these are adjusted associations, so adjusted for maternal glucose, you can see that the degree of that many of the carnitines do show a very significant association with adiposity in offspring. You can see there's actually very little association overall with birth centile, but really a strong association with many different carnitines with that skin fold sum at birth. So when we put this all together, then can we conclude that our hypothesis is correct? Well, we do know that maternal hyperglycemia does seem to prompt de novo lipogenesis. And we do know that those free fatty acids that are abundant in the maternal bloodstream do appear also to have a strong association when you adjust for glucose with body composition. We also know that there does seem to be an effect between maternal hyperglycemia on what looks like beta oxidation and carnitine abundance in the fetal circulation, and also that those carnitines appear to also be directly associated with offspring adiposity. But when we actually put all of this into our statistical models, there was still a significant effect of glucose directly on offspring adiposity, offspring body composition. And so our conclusion from that really is that there is an indirect and a direct effect of glucose. So glucose is likely to act directly on offspring body composition, but also indirectly through maternal and offspring lipid metabolism. So strengths and limitations. This was a secondary analysis of a well-regarded randomized controlled trial population. It had carefully adjudicated outcomes, and we also had a good number of cord samples. However, there is some overlap between the groups, as in the infants, of course, who developed one complication were more likely to develop another complication too. So that's just important to remember. Um, So then leading us on, really, this is heading towards the end of my presentation. But what do we then conclude? So we have talked quite a bit about intrauterine exposure to maternal diabetes and how that creates the short-term perinatal complications, particularly large for gestational age, neonatal hypoglycemia, and so on. We've shown that those perinatal or short-term complications do appear to be associated with metabolite disturbance, but particularly lipids, very pronounced in late pregnancy in the run-up to a larger gestational age baby, very pronounced from the first trimester in pregnancies that go on to deliver an infant with neonatal hypoglycemia. But we've also been able to look at the effect of whether that lipid disturbance is also likely to create longer term influences upon childhood body composition. And so our evidence so far suggests that it is certainly reasonable that lipid disturbance and altered body composition are responsible for at least some of the short-term complications, but of course may well be responsible for many of the longer-term complications as well. So just to then sum up that part, looking at metabolomics and lipidomics for us has been an incredibly powerful way to assess maternal offspring interactions in early life. We have directly caused quite a bit of controversy by suggesting that some of the perceived gospel truths of diabetes pregnancy pathophysiology are possibly not quite so clear. We have also, I think, shown really for the first time that lipid metabolism is substantially altered in type 1 diabetes in pregnancy and may have a direct role in the development of those perinatal complications. And I think these are really interesting findings. Firstly, from a clinical perspective, our population is getting steadily more obese, and lipid handling in that situation is becoming more potentially more complicated. And I think we, in the diabetes and pregnancy clinical world, need to find new ways of engaging with this and new ways of addressing these complications at an earlier stage. I'd just like to end with a few final slides. First of all, the work that we did on this study was, first of all, funded by Diabetes UK under this project grant. But also, I'm really delighted that it was selected by the American Diabetes Association as for a Young Investigator Award, and also the Pregnancy Special Interest Group Award. And so it has had a huge effect internationally, and has been hugely, in a sense, successful because of that. So thank you very much to Metabolon for all of your help with that. We're also very fortunate to have just very recently received new funding from JDRF, the Type 1 Diabetes Charity, in association with the Helmsley Charitable Trust, to do more work in metabolomics of type 1 diabetes and pregnancy. And in this work, we'll actually be looking at whether pregnancy events in mums with type 1 diabetes or women with pregnancies where there's a strong genetic risk of type 1 diabetes. We'll be looking at how pregnancy events influence the child developing type 1 diabetes in future. So again, a really important step along our understanding of how mother-child interactions influence metabolic disease risk in early life. I'd also just like to mention quickly our new study, which is called Domino, and it's, in a sense, it's another, hopefully a bigger cohort study in type 1 diabetes in pregnancy. And it's diabetes in pregnancy optimizing maternal and infant outcomes, or Domino. And we're planning, starting later this year, to recruit around 400 women from 30 clinical sites across the UK in order to look much more in depth at various aspects of maternal biochemistry in pregnancy, including insulin production and insulin resistance. We'll be looking at maternal nutrition and lifestyle choices, also looking at maternal weight management and physical activity. And this, I think, will provide a really rich resource for us to look also at differences in metabolism in pregnancy, and how we can together work through what is a healthy set of guidelines in terms of nutrition, weight management and physical activity for women with type 1 diabetes. I would just like to finish then with an acknowledgement slide. Of course, we can only do our work because people participate really kindly in our studies. So a huge thanks to all the women with type 1 diabetes who took part in CONCEPTT. It's been a study that has changed clinical care internationally, as well as changing our understanding of the pathophysiology of the disease. And so I'm so grateful for the contribution that our participants have made. Thanks also to CONCEPTT researchers, collaborators and study team, and I've put a number of them here at the bottom: Zoe Stewart, Sandra Neo, Sam Furs, Denise Feig, Albert Kuhlman and Helen Murphy. And it's been hugely it took us a long time, I have to say, to unpick some of this data, but it's been a hugely productive experience working together as a team. So thank you very much for that. And then just to acknowledge my wider collaborators, both within Leicester, within the UK and internationally. Thank you very much.
Yeah, thank you, Professor Meek, for that absolutely fantastic presentation. If I can say something, the controversy you alluded to, I think, is synonymous of groundbreaking research and truly something novel in the field. So with that, I will kick off our Q&A session. Let me see. I see quite a few questions on the right, so let's see where to begin. One question, Claire, that I see is basically, are there any biomarkers that have been selected and validated as tool for screening from this study?
Thank you very much. That's a great question. So we found actually loads of biomarkers, but our difficulty really is that because this was, or and still is, the only study in type 1 diabetes and pregnancy that has metabolomics measured, we actually don't have anything to validate our biomarkers against at the moment. And so actually that is one of the key benefits actually in our latest funding to support metabolomics assessment in the NDA study, but also of my new study Domino going forward. So we've got hopefully two opportunities on the horizon to be validating some of the biomarkers we found.
Yeah, and I would just like to reiterate that Claire is one of the amongst a handful of researchers in the world that has been granted access to this incredible cohort, which I believe has over 200,000 samples available of different nature. So I'd like to congratulate you on that, and wish you the best of luck, and look forward to working together as well. Moving
Thank you.
on. Another question really stems from the challenges around the ethical and logistical considerations regarding sample sizes and recruitment. Could you share your experience?
Sorry,
Perhaps
impact
some
of
suggestions as well.
of trying to build data sets. Well, so people say to me, how do you get people to participate in your studies? And of course, that is not, I think, the best way to approach this. The best way I think to approach this is to think, how can you design a study that other people want to take part in? And so, if you approach it from that point of view, then I think that you end up with a more appealing product, if you like, to encourage people to get involved with. So, for example, we've just finished a range of studies, but actually, it partly involves trying to understand in the pregnancy field what concerns do women really have about pregnancy? What are they most worried about in terms of complications affecting their baby? And how can I design a study and communicate that in a way that makes people realize that we are directly addressing the things they're worried about? So that's one aspect of it. CONCEPTT, of course, also offered continuous glucose monitoring, which was a new technology for women, and many women hadn't had that before. But we know it's also improved outcomes. So actually, there were major benefits for women taking part in the study as well. And I think, in many ways. We talk about sort of the ethics of studies, but actually, if women are getting better care in a study, they're getting access potentially to new technology. It's a win-win for women to take part in.
Perfect. Another question I see on the right is
Mm.
basically, were there any site-specific variabilities in the metabolomics results in the CONCEPTT study, given the recruitment was from 6 or 7 different countries?
Yes, so apologies, that's just a little bit of noise outside my window. I hope you're not picking that up too much. Just to give that a second for that to pass. That's a great question, Juan, actually. And we did see there are often differences in treatment and protocols, for example, for admission to the neonatal intensive care unit between countries, and we were very aware of this. However, we didn't feel that we had, I mean, this was the largest study in type 1 diabetes and pregnancy at the time, and probably still is. But even then, we had 225 women altogether. We had 176 who gave samples at all 3 time points. We didn't feel that we had enough to do major subset analysis with a reasonable degree of statistical robustness. So we didn't do that calculation. However, we did know that the benefits from the continuous glucose monitoring were consistent across the sites. I think there's also a lot of work to be done looking at, for example, ethnic differences in how babies grow and so forth. That's also been a very controversial issue, and lipid handling may be a little bit different across ethnic groups according to even habitual diet and so forth. We don't have the resources within CONCEPTT to do that very well either. While it was a multi-ethnic community, many of the subgroups were so small we couldn't really do that either with good statistical robustness.
Do you feel the variability in the diagnostic data can be a challenge? In reality, there are no real universally accepted set of diagnostic criteria for type 1 gestational diabetes.
So usually by the time women come to us in pregnancy, they've had type 1 diabetes for an average around 17 years in the UK. So while there are often no set diagnostic criteria, it is clear that they've had long-standing diabetes. And, almost always the data has backed up that they have had type 1 diabetes. We actually also did a little autoantibody testing on our concept cohort just to make sure that we hadn't missed anyone. And that really, it showed one instance, I think, of someone with what may well have been something like MODY. Actually, they had really high C-peptide levels, much higher than we would have expected with no autoantibodies. But actually, in general, by the time people have had type 1 diabetes for 17 to 20 years, it's following a more consistent clinical pathway compared to those early years. Um, we also have major issues actually with the diagnostic criteria for gestational diabetes across really across continents, I would say. And I think that's another issue that's made it very difficult to get a really good handle on gestational diabetes metabolomics. There's been quite a number of studies, and to some extent, they've actually shown some very common findings, regardless of the diagnostic criteria used. And I think partly, it is a continuum, this severity of hyperglycemia in pregnancy, and to some extent, while we don't know the absolute best threshold for all populations, overall, the effects of hyperglycemia do seem to be similar in terms of the metabolomics between subsets.
Thank you. I'll move on because there's quite a few questions that have come in. A question from Omar. Do you think accurate hypothesis can be created using metabolomics only? And how much of an impact did the lipidomics have in identifying further perturbations?
I think that's a fantastic question. And I agree with you. In an ideal world, there would be experimental ways to assess this that were more directly intervening. But of course, we can't do that in human pregnancy. It would be unethical to do so. And so we are in a situation where we are using the best available methods to guesstimate really what's happening. So I agree. I think there are limitations to what you can do with that. But I think also it shows us perhaps it helps us point in different directions for future experimental studies than we would have considered before. I think one of the majorly important key findings from my point of view, there had been prior to this work, there had been a belief that really glucose alone was responsible for all of the complications of diabetes in pregnancy. And actually, while we have talked a lot about lipids today, in truth, really, it's insulin deficiency that is the sum issue, that is the primary issue in diabetes and pregnancy. And of course, insulin is a master regulator both of carbohydrate and lipid homeostasis, but also proteins and so much else. Insulin is absolutely key. And what we realized having done this study is that while we thought as clinicians we were treating women well and giving them appropriate amounts of insulin, in truth, we weren't matching the insulin well enough to dietary intake and so forth, that there was still a degree of insulin insufficiency on board, really, that was contributing towards these complications. So I think it really encouraged us to go back to the clinic to think through how we actually deliver insulin in diabetes and pregnancy, whether we could be doing that better, how we assess optimal insulin dosage, and so forth. With increased changes recently in terms of care pathways through what's called hybrid closed loop development, so an artificial pancreas situation, we actually are now able to look at that in much greater depth than we ever have done before.
And I think we have some time for a few more. Is CGM, so continuous glucose monitoring, better than measuring HbA1c in gestational diabetes?
Well, so that's a great question, and I'd love to know the answer as well. But unfortunately, that is still a field that is in its infancy, relatively speaking. Um, In my view, I've studied HbA1c quite a bit actually in gestational diabetes. In my view, HbA1c is excellent for identifying, for example, pre existing type 2 diabetes undiagnosed, pre existing pre diabetes. And those groups of patients are often kind of hidden within the gestational diabetes population. Um, and so HbA1c, I think, can have a really useful role, especially in early pregnancy and gestational diabetes. However, continuous glucose monitoring, there's really two studies that have recently been done, large scale studies looking at continuous glucose monitoring. They've not been published yet, but they have been presented internationally. And those show that actually continuous glucose monitoring is able to show, even from early pregnancy, predictive markers, really predictive levels of glucose that predict later gestational diabetes. So the answer to that is not entirely clear yet. There is clearly some association with glycemia in early pregnancy, but we, as a diabetes and pregnancy community, still don't really know the answer to that question.
Thank you, Claire. And I picked a question that can help perhaps convey the interaction that Metabolon has with its clients when it comes to grant writing. So, what level of service does Metabolon offer? What was your experience when it comes to our support to acquire the grants?
Super. So, we do a lot of grant writing, and there's a range of different kind of approaches, I suppose, from collaborators that we've had in the past. But really, at the stage of designing the study, it's my understanding that Metabolon are happy to be involved, often give input into the study design itself, and are willing to supply, preliminary data and/or information to support that part of the methodology section of the grant. Certainly, that worked very well for us in the recent grant that we've got from to use with the India consortium. And so, going ahead, if I was considering another grant, then I would probably get Metabolon involved at a fairly early stage.
Thank you. Let's
Mm.
see, perhaps a couple more, and then I'll try and wrap it up. Um, how can we prevent hyperinsulinemia in fetus, which is a main precursor for adult onset of like type 2 diabetes and visceral adiposity?
Yeah, so I think this is a fantastic question. And again, if I knew the answer to this, all of our problems would be solved. Um, so, to some extent, we can prevent fetal hyperinsulinism by controlling maternal glucose. And that is, in a sense, still the sort of take home message for clinic, that anything that you do to bring maternal glucose down to a healthier level will have benefits for the baby. However, there is a degree of fetal response that is probably to some extent genetically determined, possibly influenced by other factors such as maternal diet, obesity, many other things that seem to influence the degree of hyperinsulinemia that each individual fetus may get. We still have, again, very little real understanding of that. It's a very difficult situation to study. In my view, there seems to be something happening mid-pregnancy, and again, that is from these results. Really, there's something happening mid-pregnancy that seems to change that trajectory of hyperinsulinism, if you like, hyperinsulinemia. That means that some babies, despite having comparable levels of glucose, some become much more hyperinsulinemic than others. We still don't really know why. And certainly we're targeting that area in pregnancy really carefully with our future studies to make sure that we're trying to identify some of those pieces of the jigsaw.
Thank you. And I'm going to take this opportunity to use you because I have strong feelings about this. What advantages do metabolomics and lipidomics give that other omics don't offer?
Well, as a clinical biochemist, of course, why would you go beyond metabolomics and lipidomics? No, I think, we know that there are obviously genetic aspects to diabetes in pregnancy, but actually, we also know that what happens in utero in this situation is almost certainly predominantly metabolic rather than, if you like, the effects of purely genetic susceptibility to disease. So I think, the metabolomics and lipidomics allows us to study that really well. And you alluded a little bit to this earlier. The other thing that we were able to use the metabolomics to look at was to gain much more of an understanding of maternal dietary composition. It's really difficult to study diet well longitudinally. We use a 24-hour recall to do this, and we'll be doing this much more in our study Domino coming soon. But actually, trying to find reliable markers of maternal dietary intake would actually be hugely helpful going forward. There's not a lot of information actually on optimal diet in diabetes and pregnancy, and that's an area I'm really keen that we clarify.
Thank you for that, Professor Meek. I'll put one last question. Do we have any data on effects of placenta metabolomics on offspring adiposity? Um, the research findings of the person who posed this question show maternal obesity had more adverse effects on placenta function compared to GDM.
Fantastic. Hi, Ezekiel. It's lovely to hear that you are there. Ezekiel is a fantastic placental researcher who now works in diabetes care in the north of England. So the answer is that there have been studies, not necessarily on placental metabolomics, but we actually did some placental lipidomics as a method development in house a little while ago. And yes, you can certainly study placenta in terms of metabolomics and lipidomics. We don't have any data really in the type 1 diabetes world in placenta at the moment from our studies, although we will be trying to establish much more of a kind of placental bank, sample bank going forward that will allow us to look at this in much more detail.
I lied. I want to give room to one last question because I think it's important for the researchers. How do you find collaborating worldwide? So, EU, US, Australia, and what advice would you give to other researchers wanting to work with cohorts and biobanks in different countries?
Fantastic. So, I mean, it's one of the most fun parts of my job, I would say, talking to and getting to know people all around the world. Having said that, I think collaborations are generally easier if you've had a little bit of face-to-face contact at some stage. And so I think, international conferences are really important. Just starting to get to know people within the field and having those corridor chats, if you like, that lead to really good collaborations. I think, as with all collaborations, international or national, there are differences often in working styles, timekeeping, all of that, that actually can create challenges. I think it's useful usually to be fairly clear in advance as to, what you're offering, what you can do, and what you can't do. Having said that, I think there's huge opportunities. And for example, the study that we're just about to embark on with the Australian collaborators with the NDA study, we've had a number of very useful meetings with them, really, excuse me. And it's just been clear from the very beginning that we've actually had one, a common desire to understand type 1 diabetes, and two, just a real desire to make sure the samples are used really well, really constructively for patients, but also for the research community.
It's good to hear that science can go beyond boundaries, if I have to be honest, especially in this time. Um, Professor Meek, Claire, I would like to point out that there is a lot of thank yous that have come in, and a lot of people have congratulated you for this presentation. So hopefully we can repeat this experience in the near future. I would like to thank you again for this availability. I've been a very proud host, so thank you again. And I guess we look forward to seeing everyone on our next webinar. So with that, I wish everyone a nice rest of the day wherever you are in the world, and see you next time. Thank you
Thank you
again.
very much. Thank you.
Bye-bye.